Dun, 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 dun. Hey, it's me again. So just checking in, see how you guys are doing. Also thought we could talk about DNA replication today. So as we talked about in class, again, this hopefully should all be a review. DNA replication is when we take our double helix and we separate it and use each of the original strands as a template to build the other. Now, the reason why we want to do this is simply because anytime you make more cells through mitosis, you need to put more DNA into those so they have the instructions to do whatever the cell needs to do. So if it's a stomach cell, it needs the instructions to make, enzymes and acids and be able to digest and all that. If it's a brain cell, to make the neurotransmitters. So today we're going to talk about how exactly it is that we replicate our DNA. Now for my bio kids, we're going to talk about one extra enzyme called Primase. We didn't talk about that in class. It's fine. Don't worry about it too much. It was when we talked about my genetics class. And my genetics kids, everything else should be the same. All right, so first thing we have to remember is that DNA is anti-parallel, meaning it's going in two different directions. So we have, you ready for this? We have the 5' end there and the 3' end of the other strand. Then 5, 3, and 5. So what those 5 and 3 refer to are how the sugar molecule, the deoxyribose, is actually orientated. The 3 and 5 refer to particular sugars and which way they're facing. So if you're looking this way, the 5th carbon is facing that way and the 3rd carbon is facing that way. Now, since we know that the other strand is opposite because it's upside down, that means that the 5th carbon is facing that way and the 3rd carbon is facing this way. This is actually really important because one of our enzymes, Polymerase, can only attach and read and move in a certain direction relative to the deoxyribose sugars. We'll get to that in a moment. First thing first. Now, DNA, it's twisted, it's bonded in the middle with hydrogen bonds. So first thing we need is an enzyme to come in and untwist it. It's going to come in, it's going to actually open up the double helix, and it's going to break the hydrogen bonds between the two strands. Now this particular enzyme is called DNA helicase because it opens up and unwinds the double helix. So we have DNA helicase. Not too bad so far. Now the point where it's at is, we call that a replication fork, kind of like a fork in the road. You're going in one way and then there's two options. So what happens is we have our double helix and then it splits and opens up. Now something to keep in mind, this helicase is going to be moving that way so it can keep opening up and unwinding more and more of the DNA. Now, this is the enzyme that my bio kids haven't learned about, but my genetics kids have. It's called Primase. Now, what Primase does is it's going to build... That's kind of hard to see. I'm sorry. Bear with me. It's going to build these little things called primers. Now, they're RNA-based. They're just a few bases long, usually. And what happens is it provides a launching point for our next enzyme, the polymerase. Now, as you might know, otherwise we're going to get to it later, RNA is also nucleic acid, not the same as DNA though. It does not have a thymine, it has uracil. It also has a different sugar. We will get to that in a later video. So, it provides us a little base. That's our primer. Our RNA primer provides us a base that the next enzyme, polymerase, can actually come in and work off of. So our next enzyme, hi Ripley, is called DNA polymerase. Now remember, DNA is a macromolecule, so it's a polymer. Polymers are made up of a whole bunch of monomers bound together. So what polymerase is going to do is actually going to read the parent strand, and it is going to, you're a good boy. It's going to read the parent strand, the original DNA, and it's going to attach the complementary bases relative to the original strand. So I'm going to have DNA polymerase. That is DNA polymerase. And what it's going to do is it's actually going to move in this direction, because how polymerase can move, remember when we were talking about that 3 to 5? Polymerase can only move in a 5 to 3 direction. So in the original strand, we have the 5th prime end here and the 3rd prime end here. That means when it builds the new strand, starting here, that's going to be the 5 prime end of the new strand. Because remember, top and bottom strands run opposite each other. Yes, I know. They run opposite each other. So it's going to attach to the primer. That's going to be its launching off point. It's kind of like those old Wile E. Coyote and Roadrunner cartoons. Really, dude. So what happens is, in the old cartoons, you'd see Wile E. Coyote standing on a cliff, and he starts building, a ledge or something off it using 2x4s, but he keeps taking the previous one to make the next one, and suddenly he's floating in midair. You can't float in midair. You need a launching point. So that's what the primer is. It is simply a launching point for the polymerase to attach to in order to start building off of. So it attaches to the primer, and then it's going to read the parent strand. Now, polymerase is going this way in a 5 to 3 direction. It builds 5 to 3. It doesn't read 5 to 3. It builds 5 to 3. So it's going to keep following the the helicase. It's going to keep following the helicase. And as it reads the original strand, it's going to be attaching nucleotides with complementary bases. So if there's a C underneath there, it would attach a base with G. Yes, you can admire my Rugrats PJs. T, so A, T, G, that's supposed to be a C. A, T, A, C, G, C, G, T, T, A, G, C. So it's just going to keep going right behind the DNA helicase, making this the 3' end because that is the 5' end. Now we call this the leading strand because it can just keep following on and on and on behind the helicase. So it's just nice and continuous. However, we can't do that same process on the bottom strand because remember, top and bottom opposite each other, which means we cannot build 5 to 3 going this way. We have to build 3 to 5. And that ain't gonna work. Simply put, to go this way, we'd be building 3 to 5. And polymerase cannot work in that direction. So to move in the 5 to 3 direction, it actually has to, how best to put this, leapfrog. It's like that old childhood game. What I mean by that is we're going to have quite a few more primers. So when the helicase was around here, a primer was built here. I'm just going to go ahead and draw a few more primers. It just keeps adding them. So those are all the primers, the RNA primers that were built by primates. Now what happens is polymerase is going to attach to this one because this one was the one that was added first. and it is going to start building in a 5'3" direction. Now again, because this parent strand is opposite the top one, that means it cannot go this way. It has to move in that direction. Now that seems counterintuitive until we realize, hey, leapfrog. So it's going to attach here and start building in this direction. So we have C, A, T, T. When it gets to a point that it can no longer build any more bases, it's going to detach from the strand and move all the way to the next primer. And then when it attaches to that primer, it's going to start working backwards again. So we have T, A, G. That's part of the primer. And then when it gets to that point, it can't build any further because the primer is there. It is going to detach and leapfrog ahead. Go to the next primer. And start building backwards from there. So we have T, A, C. Now, because of all the attaching and detaching and moving around, this strand does take a little longer to build, so we call it the lagging strand. Now, the lagging strand, you notice there's a lot more fragments than there are in the leading strand. And we call these fragments Okazaki fragments. Now, what has to happen once the strands have been completed, and get rid of some stuff. Don't worry, I'm going to redraw a few things. Let's see, we have our DNA. Need the primers. So we have a primer there, and that is A and U. And we have a primer here, which is G, C. And we have a primer here, which is also G and C. So pretty much I just wanted to take off or remove the enzyme so you can see this a little bit clearly. Now, what we have here is DNA is in the red, that's the new strand, and these little light pink areas that did not show up as well as I thought they would, those are the RNA primers. We cannot leave RNA nucleotides in a DNA molecule. So what has to happen is they have to be cut out. So an enzyme is actually going to come through and cut those out. Well, that leaves gaps. So what do you think has to happen? Polymerase has to come back. And it actually has places that it can attach from now. So it can attach and move backwards. G, C. Let's see. A, T. And in all reality, there's also a helicase here doing the same thing in that direction. So there would be something for it to attach here as well. And this helicase would have kept moving, so another primer and Okazaki fragment would have been built as well. All right. So now we have our DNA. But if you notice, it's still not connected. So that's where our next enzyme comes in. That one is called ligase, DNA ligase. And what DNA ligase does, a little enzyme, I mean, technically they're all little because they're on a site, or, submolecular level. But what happens is it's just going to actually go along the backbone and find any place where the sugar phosphate backbone is not connected. And then it is going to fuse them. So that it becomes continuous. Now, DNA polymerase does come back for one more job, it's actually going to do proofreading. So it's going to scan the new strand that it has made, and it's going to look to see if there's any errors. If it recognizes that there's any errors, it will cut out that section and replace it. Now notice what I said. If it recognizes that there are errors, there are times where it's not recognized and the error goes unnoticed until the next step, which would be protein synthesis. But we'll get to that in a later video. We still have a protein synthesis video, and then we'll get to some mutation videos. But once we're all done, the two new DNA helixes, top and bottom, will separate and wind back up. And now we have two DNA double helixes in your cell, and it is all set for the cell division when we start making another cell, and now we have DNA to put in that cell. Now, something I do want you to keep in mind is this is not going on at just one single place. Our genome is too big. The eukaryotic genome is too big. It cannot just start at one place and go all the way through. It can in prokaryotes, bacteria, but not in eukaryotic DNA. So what is going on is this whole thing, which, by the way, we only showed this side of it. We didn't even show that side because there's a helicase there moving in that direction, and we have replication going on there too. But this is going on at hundreds of places across your DNA all at once. That's the only way that we're able to make copies of our DNA in an effective, timely manner. So it's going on at hundreds of places all at once, which means even this guy, which looks like he's continuous, not truly continuous, because eventually he's going to run into a spot where there's already, another replication fork down the road, and he's going to have to be fused up there. So ligase will have to fuse up those segments. So it's a really, really, really complex process that occurs hundreds of places around your DNA all simultaneously. And I know my kids have heard me say this numerous times in class, but isn't nature cool? I mean, the fact that it can do all this and check for errors and fix those errors, that's pretty amazing. All right, so there's that. Until the next video, there'll probably be one more today. We'll see. Maybe that'll be the cell cycle. Haha. But it's either going to be cell cycle or protein synthesis. We'll see. But until next time, take care, stay healthy, and know that Komar misses you. So take care. Bye-bye.